So I'm going to follow on now from a wonderful talk that Yoni Nazarati just gave before lunch, where he's laid this wonderful foundation about transformer models and attention mechanisms and how that all works. Because what I'm going to talk about is foundation models in agriculture, and that really does build upon the transformer model most of the time. So let's just talk for a second about traditional modeling. We probably a lot of you here have got a data set and fit whether it's a regression model or a machine learning model like random forest or XGBoost or something. And typically you have a whole lot of data and labels. This is what's called supervised learning. You have labeled information and you train a model, and the model could potentially fit that data very well and might be really good at making predictions for that specific kind of data. But the problem is that these models, really, they're designed for that one task, and they generally don't transfer well to doing anything else. And not to mention the fact that getting good data and good amounts of that data is difficult. It's expensive often. Like you might have to do a lot of phenotyping, a lot of assays for your crop or livestock or whatever it is you work with. So another type of model is a foundation model. A foundation model is this definition here, a large model pre-trained, usually with self-supervised learning on broad, unlabeled data. So if we compare that to in the previous slide where you got tables, the blue model and the orange model, you have tables of labeled data. So this would be a supervised training exercise. With the foundation model, you don't have that kind of data. The model has to kind of teach itself in a way from the data. It has to figure out what's in the data on its own. But once you have built a foundation model, you can then adapt it to many downstream tasks. You can fine-tune it so that it can do all sorts of different types of predictions and lean on what was learned at the foundation level. So in an analogy, you can imagine if you've laid the foundations for a building, a big slab of concrete perhaps in the ground. On top of that, you could build a house, an office, a garage, all sorts of things. But because you have the foundation there, you can build on top of it. Without the foundation, you couldn't build any of those things. So it is with foundation models. Now, foundation models have been trained and exist across a lot of biological layers that most of us here are familiar with, from right down at the bottom level where we have DNA, up through RNA, protein, to phenotype and the environment. And what it means is that whichever level you might be working with in your data, there are these large models that are trained to understand information at that level. And I've provided here just a bit of a snapshot of some of the existing models that are already trained and already out there for our use at each of these levels. So I'm going to spend most of my time explaining the bottom one, the DNA level, foundation models for DNA. I do a lot of work with DNA. I do a lot of work with SNPs and structural variation and genotyping and the relationship between those and phenotype. And so that's just where I'm most familiar. But you might have heard of some of the protein models, which model protein structure. And I will also touch upon the top layer, environment foundation models towards the end, which I think is a really interesting aspect and might be very useful for a lot of people here. So Yoni has explained how a model like ChatGPT works under the hood. And really, a large language model like GPT is a foundation model for language. You have the phrase, the quick brown fox, and the model understands the structure of the language. It's been trained to understand structure and grammar and the things that matter within the language. So it can reliably predict the next word. And it does this, and though Yoni's kind of shown this already, I'm going to do a quick sort of review again. It does this by tokenizing, in this case, the words the quick brown fox. And those tokens pulled out of a dictionary. There's a dictionary of all the tokens. And each one has a meaning, and that meaning is conveyed in an embedding. So we have an embedding for the, quick, brown, and fox, 4 embeddings. And remember, these embeddings are often quite a large dimension, and they're a vector of numbers. And then those pass into the attention heads, the attention system, which I've just called matrix magic. Yanni was able to explain the matrix magic, but what you end up with is this output of attention scores, how each token attends to each other. And that conveys not just now the meaning of each token, but the context, the meaning within this input. And so what you end up with is on the end there, these colorful representation of these embeddings. The embeddings have been transformed from just the meaning of each word or each token to a contextual meaning. They are context-aware embeddings. And then those go into the prediction mechanism, and the model gives you a probability distribution of what's the next word. And in this case, of course, the highest probability would be jumps. That doesn't mean that the next word will always be jumps. These are drawn from a distribution. DNA is just a language with a different vocabulary and grammar. So the architecture you see here in this transformer model works with DNA just as it does with English or any other language. In the case of this little input string up there, CGA, TCG, etc., it can be tokenized in many ways, but in my little example, it's tokenized into freemers, so little chunks of free DNA nucleotides, and each of them has an embedding. And each of those then goes in and gets attended to each other, and you get these context-aware embeddings of the DNA. And the idea is that the model can predict what's next. And it can do that because it's been trained. It was trained on millions and millions and millions of sequences of DNA. So if you were to train a DNA foundation model on human DNA, you would grab many human genomes and split it up into chunks and feed this in. And what you would do to make this an unsupervised, sort of self-supervised learning task, you would mask out, and I think that's my next slide, yep. You mask out some of the DNA, and then the model's job is to try and predict what's missing. When it gets it right, that's great. When it gets it wrong, that's a calculation of that is your loss. How right was it or how wrong was it? If it's wrong a lot, that loss is fed back propagated through the model during training, and the weights of the model are tweaked up or down, and then you do it all again many, many times until the loss is minimized. The model becomes very good at predicting what's next, what's missing in the DNA, and then the weights get fixed. And what you have there in that model and in those weights is this intrinsic understanding of how DNA works. It's like understanding that when you see certain patterns when certain patterns exist in the DNA, those relate to each other. And we ourselves as humans, we can't do this. We can't look at a long stretch of DNA and see those patterns. But a model that has, say, 1 billion parameters, all those weights between the neurons in it, and these attention mechanisms, it can. And so that's what builds us this foundation model of the understanding of DNA. And we then can lean on that and use that information to our advantage. So what advantage? What's the point? Yay, now I can chat with DNA. It's not going to be the most scintillating conversation, right? So what can you do with one of these foundation models? Well, what matters, this quote here, sufficiently large DNA models that are trained on diverse genomes acquire generalizable representations that support a wide range of downstream prediction tasks. And they can do this in sort of 3 main ways. They can do this what's called a zero-shot prediction. They can do it through embeddings, or they can do it through fine-tuning. And I'll go through each of these now. Zero-shot means really that what the foundation model was originally trained to do was predict what's next, either what's missing or what's next. And if we mask out a piece of our DNA, we'll get a prediction at the end of the foundation model of what's missing. And that actually can be useful for downstream tasks. You might think, what sort of task do you need to know what's missing? It's not about necessarily what's missing. It's about the probability of what's missing, because you can start calculating ratios between different femurs, in this case, different Kainers, which ones are more probable than others. And that can tell us something about the evolution of that DNA, the importance of different Kainers within that stretch of DNA. We can also look at the embeddings. So remember, we have each token has an embedding about what it means. In this case, what a token means is very esoteric, not like a word like the quick brown fox. What does CGA mean? Doesn't really mean anything to me and you, but it might mean something to the model. I've just got a question at the back there. I'll take that. Yeah. So the question was, does meaning mean context in this? In the blue set of embeddings there that are at the start of the model, those are more about a non-contextual meaning. This is a bit harder to explain in terms of DNA than it is in terms of English. The embeddings that have gone through the attention system and come out on the other side that have been transformed, those convey context, whereas the original embeddings just contain a meaning. As I said, it's hard to explain in English what that meaning might be. What is the meaning of CGA? To us, not much. To the model, there is intrinsically a meaning to that. It's very hard to interpret that in English. But the idea is that at the end, you have these transformed embeddings that conveys both the meanings of those tokens and their context within the entire input that you gave. And so you can average that set of embeddings, and now you get one single embedding that represents the entire input sequence. So we now have our extract and embedding, and we can do really cool stuff with embeddings. We can take two sequences that are really quite different, sequence A, sequence B. We can get an embedding of A and an embedding of B. And as Yoni was talking about, we can look at the distance between them or the similarity of them using a mechanism like cosine similarity, right? So there's an angle between embeddings, and we can look and see if they are near to each other in this big multi-dimensional space, or if they're far away from each other. And that can tell us something about the relationship between these two stretches of DNA. To put that in practical terms, here's outputs. This is a figure from an article about hyena DNA, which is a DNA foundation model. And they were comparing three different foundation models: hyena, nucleotide transformer, and DNA BERT. And they got embeddings of sequences where they already knew where that sequence was coming from. So if the sequence is from a protein coding region, or from long non-coding RNA, or from microRNA, and all these kinds of different genomic types. And then if you cluster those and bring it down to a two-dimensional embedding space, so you've basically dimension reduced from a very large embedding down to very small, you can see that color-wise, similar types of DNA cluster together. So that tells us that the embeddings mean something. They're capturing meaning with regards to what type of DNA, what type of genomic feature that DNA is coming from. They capture way more than that, but that's all that this plot is designed to show. Now we can apply this concept to SNPs. I'm not sure if does everyone here work with SNPs in some way? Anyone? Single nucleotide polymorphism. So, they're the basic unit of breeding and selection. That's your basic genetic variation is usually captured in SNPs. With embeddings, we can basically take the same stretch of DNA and alter it at just one nucleotide to represent the reference sequence and the alternate sequence. And here you have two sequences that are virtually identical. And we might be talking about, say, a 1 kilobase region with one change. You'd expect that since these sequences are almost identical, that their embeddings would be almost identical, and there'd be very little difference between the two vectors representing their embedding. But every now and then, you find that the difference between the reference and the alternate sequence diverges more. And that tells us that that single change in nucleotide, according to the model, matters. It's changing the model's perception of that sequence of data just by changing one nucleotide. Its internal understanding of that sequence of DNA has changed. And so that therefore means to us that that nucleotide swap, that SNP, has some sort of impact. It's hard to nail down exactly what that impact is on. It doesn't refer to a specific trait, but there could be some functional consequence of that change. And so we can use this system to rank SNPs by impact. And on day 3, for those of you who are attending the workshops on day 3, we'll actually do this kind of scoring mechanism of SNPs hands-on in that workshop, and show how you can take a SNP set that's measured in your population and rank the SNPs according to this scoring mechanisms like this, and use them for optimizing genomic prediction models. Another way that you can use embeddings is more directly as input to machine learning. So as I showed in the beginning, the traditional machine learning model, you have some trait that you're interested in, you measure a bunch of features. And we saw in an earlier talk today with the iris data set, where you have a few features like the sepal width and the petal length and things like that. And that can predict the class of iris, the species of iris. And that would be a standard machine learning model. Well, if you can get embeddings, then you can basically have your input come in from your first sample, get an embedding, and load that embedding up as features of the model. So if we're talking about, say, a 1,000 length vector for an embedding, that's the equivalent of having 1,000 features in a predictor matrix for your machine learning model. And we can then do sample 2 and get its embedding, and all the way down to sample n. And what we built is a feature matrix without having to have really necessarily measured anything else. Just from the DNA here, we've now built this feature matrix, which captures more than just the string of DNA, but everything the model knows about that DNA, all the structural context, anything to do with how that DNA might affect other stretches of DNA, might affect expression and genes, and all of that is intrinsically captured during the training of the foundation model. And so now this structure can be used as a really powerful machine learning model. And then the third way to use foundation models for predicting things is through fine-tuning. Fine-tuning means taking a foundation model and tweaking it so it's now a little bit better at a specific task than it would be if you just did zero-shot or embedding-based prediction. So as an example, imagine we have a whole bunch of DNA strings that are from promoter regions. We generally know that promoters are where a lot of the control mechanism happens for gene expression. And so if we've measured gene expression of a whole lot of genes and we've got the promoter regions, we could build a model of how DNA predicts gene expression. And so what we could do is we could take our foundation model and run it through a fine-tuning mechanism. And that means that we're adding a new predictive head, in this case, an expression prediction head, onto the end of the model. So we've got the foundation model and it's got its architecture in there, as Yoni described, tension mechanisms and all sorts of layers of neural networks and architectures going on. But we can add on the end a special prediction head that's designed just for predicting gene expression. And so the promoters go in, the DNA goes in, the expression head predicts a value of expression. And then depending on how right or wrong it is, the model gets trained and tweaked, and the weights in the expression head get trained to predict expression given DNA embeddings. But also, you'll see that in the blue area, where there were a whole lot of layers that exist in the foundation model, the last layer I've turned into kind of a greeny blue color. That represents the fact that we're not just training a new expression head that's tacked onto the end of the model, but also some of the weights of the original foundation model, right towards the end of it, also get tweaked. They can also do some learning in that training process. And so what you end up with is kind of a modified foundation model that's tilted or biased towards predicting gene expression in this example, coupled with a specialized prediction head that's entirely geared towards predicting gene expression. And with it, you have a fine-tuned foundation model. And so now you can take any stretch of DNA, put it in there, and get a gene expression prediction. Fortunately, for many of the foundation models that exist out there, they often have fine-tuned versions as well. So here's an example in the plant space. This is a foundation model called AgroNT. NT stands for nucleotide transformer. It was trained on about, I think, 48 species of plant from across a wide variety of clades. And you can see in panel B there, this is a figure from their paper. They show that whole masked training system. And then in panel C, they show the fact that you can fine-tune, AgroNT can be fine-tuned, add a new prediction head. It's got some rescaled weights and produces an output. Okay, so that's the whole process I've just been describing. Another competing one in the plant world is called PDLLMs. That stands for Plant DNA Large Language Models. So they trained a whole bunch of different ones using different architectures like BERT, GPT, NT, Gemma, and Mamba. And what they've done here in this table is they've shown how accurate these different fine-tuned models are. They've fine-tuned these models for a whole bunch of different tasks, basically trying to predict genomic features from raw DNA inputs. So each row represents a fine-tuned task, and each number represents the accuracy that each model got in trying to predict that feature. So if you look at the top row, our models here were fine-tuned to predict whether a stretch of DNA is a core promoter or not. It's a classifier. And the accuracy ranges from about 90% with the GPT model, shown in red, down to about 77% for the DNA BERT2 model. And you can see that for any given task, the accuracy varies drastically depending on the architecture of the model. They've all got different parameter numbers, different systems. And so there's no one model that's best at everything. For PDLLMs, they're a smaller, more compact model than AgroNT. That's one of the benefits of it. There are quite a few DNA foundation models available for plants. This table shows some of the differences. Like AgroNT is about 1 billion parameters, or something like the plant CAD2S, which stands for small. That model is only 88 million parameters. In the workshop on Wednesday, we'll actually be using the plant CAD2 small model. So you'll actually get hands-on with that model, and you'll learn how to access these models, download them, run them in a script, and actually get embeddings of DNA and use the features that they provide. What about animals? I know some people here are not from a crops background, but more working with livestock, cattle. Well, there are various foundation models that are trained on mammalian DNA, and I've listed 4 here. Again, you'd need to kind of read the papers to understand which ones are best for the species that you're working with. So one of the great things about these models is you can actually do cross-species stuff. So if I were to take a model like AgroNT, which is trained on a whole bunch of species of crops and plants, but I'm working with one that's not in that training set, as long as some of the plants in that are phylogenetically reasonably close, I can probably make some pretty good predictions with the DNA from my species that the foundation model was not even trained on. There might be a bit of a loss in accuracy, but actually the ability to transfer learn and do cross-species generalization is super powerful. But sometimes foundation models are not the best model for everything. Sometimes if you have lots of labeled data, enormous amounts of labeled data, training a dedicated model might do better than a foundation model for prediction tasks. So I'm just going to finish up quickly then in the last minute or two, talking about environment level foundation models. I've suggested three here: Prithvi, Clay, and Alpha Earth. Alpha Earth's put out by Google, and what they've basically done is they've combined satellite imagery, environmental data from all across the globe at a really small resolution. So I think it might be down to 10 meters squared resolution or something like this. And they've taken all that data and generated embeddings for every spot on the planet. And the embedding, in their case, it's just a 64 dimensional embedding. So quite small, quite compact. And so any space on the planet, you can give it the input as a latitude and longitude, and you can get this embedding back, which represents everything the model knows about the environment at that spot, given by all sorts of different satellite imaging, NDVI, greenery, drought stuff, crop information, and whatever else. I forget what's gone into that model. And so that's incredibly powerful, because then what we can do is we can take DNA and get our embeddings of DNA for a sample. We can also take the latitude and longitude where that sample came from. And we now have a combined genotype and environment embedding. And those can be all used as features in machine learning. So now we've gone beyond just having genetic data linking to a trait. We've got genetic and environmental data at the same time. And you know what? We haven't had to do any extra work to do it. We just have to extract it from an already existing foundation model. So with that, I'd like to say thank you. And happy to, I think we've probably got a couple of minutes available for questions. And yes, for those in the workshops, you'll see more on foundation models on Wednesday.
Thanks for the talk. Just a quick question. Do you ever have to do trickery or wizardry with such long, because obviously entire strings of DNA are massive. Is there any finagling that you have to do to allow the tricks with the attention of these models?
Yeah, so each one has a limit on the size of its context. So on the right column, on the last column here, see that max context length? That's the limit of attention. So the largest one here is 1 megabase. They can take because of the architecture specifically of that model, it's able to take 1 megabase in. Whereas some of the others are very short context, like 512 bases. So it very much depends on the model you're using. To be more specific, if you're going to put in a megabase of DNA into like the NT system, you can expect that context kind of does decay and get worse as you put in such a big chunk of DNA. If you have a model that can take 1 megabase, you don't have to put a megabase in. You can put much smaller. So I hope that answers the question.
How does the ploidy level affects these predictions?
How does the ploidy level affect what?
Affects predictions of these models?
That's a really good question. During training, the model is likely to have seen, if multiple genomes are put in, it's likely to have seen the effects of the ploidy and the variance that it's seen between the varying chromosomes, and it will know that certain spots are more like certain spots in the DNA are more affected than others, less conserved across the ploidy, if you know what I mean. But I believe that really polyploid stuff requires usually quite special training, and I think there are some foundation models being built specifically for highly polyploid species. Yep.
Thank you.
Previously, you said you can input a sequence of gene and you predict the expression of this gene, right? But we know the expression of gene also can controlled by another genes. So if we just input one sequence, how we know the background?
Yeah. Now you've kind of I'm busted here. Of all the things that these foundation models can predict, gene expression is one of the hardest because of all the other things that can affect gene expression. Nevertheless, when it's trained, the foundation model, when it's pre-trained, it's learning those relationships between regions of the DNA, including the genes themselves. And so some of that sort of epistatic and long-range relationships is under the hood being learned and being applied. When it comes down to the prediction stage, some of those learnings in the model are being applied. But nevertheless, it is been shown that the accuracy of prediction of gene expression itself is not particularly strong. Some models are stronger than others. But I use gene expression as just one example. Like I showed, some of these things have much like predicting things like conservation of DNA, very, very strong, predicting methylation states and things like this are much stronger. Gene expression, not quite.
Yeah, because they are conventional, they have some history. Yeah.
I think one more.
Hey David, thanks for
Hey.
the talk. My question is like from my understanding, the foundation models can't. You also mentioned in your slides like they can't beat the special models in the specific area. So my question is like, do you have like any thinking about like why the foundation models can't beat them? Like for example, in this table, I think all of the listed baselines are all like LLM-based, transformer-based models, but there's no like machine learning based baselines. So why?
There are if I haven't shown them here, but in most of the papers for these models, they do show against usually the state-of-the-art machine learning models. Sometimes they can beat them. It depends on the task. When they don't beat them, I think it's that the foundation model provides a very generalized view of the DNA, and a lot of the learning that the model has done and the parameters in the weights are not geared towards that final task. And even with fine-tuning, you're still leaving the vast majority of parameters learned for general understanding of DNA, not for specifically understanding the relationship between these two things. Whereas the dedicated machine learning model is purely designed to learn the relationship between those two things. So it's kind of got a little bit of an advantage there. It's dedicated to learning that relationship. And so the problem is that the dedicated models, you usually need a ton of label data to do it well. The foundation model, you don't need that much data to fine tune it, and you can still get most of the way or all the way to the same level as a very dedicated model. And you still have the diversity of the foundation model to work with.
Maybe following up, like from my understanding, like current cloud or ChatGPT models, they can even beat the special models in like almost all the time. So that this means like we don't treat the gene foundation models with enough data.
We don't.
Like for example, like GPT-5, it can beat the specialist models in lots of like different areas. So that means that
I mean, potentially, but GPT-5 we can't go and fine tune or use ourselves. Like we can't download that and use those ourselves. Yeah. We have limitations on how we can use something like GPT-5. But I think we'll have to leave it there. But I'll have a chat with you after